The overall goal of this filter insert-based 3D culture system for patient-derived primary cells is to establish a more biologically relevant in vitro model system for prostate cancer research.
By using primary human cells, this method can help answer key questions in prostate developmental biology as well as in prostate cancer. The main advantage of this technique is that it is a relatively rapid in vitro methodology for the establishment of differentiated primary prostate cells, which also allows for the rapid isolation of biomolecules such as RNA, DNA, and protein.
Visual demonstration of these methods are critical as the processing of cells on the filter insert for histology or for RNA and protein collection are delicate and time sensitive.
To help limit diffusion between the chambers, in a biological safety cabinet, apply a thin layer of 0.1% gelatin to the bottom side of the inserts and allow them to dry. Repeat this procedure 2 more times. Then, apply the cells to the inner chamber of the gelatin-coated inserts and culture them for up to 2 weeks. After 2 weeks, apply the cultured filter inserts directly to one of the appropriate downstream applications detailed in the followings. To begin this procedure, aspirate PBS from the outer chamber and carefully remove PBS from the inner chamber. The cultured inserts can be kept briefly in PBS until the application is ready to begin, but do not allow the membrane to dry out. Next, add 100 microliters of 0.25% trypsin EDTA to each inner chamber. Then, incubate them for 5 minutes at 37 degrees Celsius. Afterward, briefly and carefully scrape the cells on the membrane surface.